One of the major questions in biology is that how cell specification and differentiation occur in embryo development. Transcription factors that are involved in embryogenesis and organogenesis are conserved during evolution. In our lab, we are using Drosophila embryo to study early stages of embryogenesis. In the early embryo, maternally loaded transcription factors are driving the early zygotic genomic activation. Previous studies have shown that right before gastrulation, two other transcription factors like OPA and OC start being expressed and are driving a second wave of zygotic genomic in different parts of the embryo. However, it is still unknown that proteins like OPA have a role in cell specification, or is their role for OPA similar to OC in head development. In our recently published paper, we also discussed the potential role for OPA in head development. Here we can see that our in situ data that is showing that OPA and OC are overlapping in future head domain before gastrulation, and we can see that the overlap is about 12% before gastrulation, and this number drops down to 3% after gastrulation. We using super resolution microscopy, we again noticed that before gastrulation the overlap was about 2 to 4 cells, but after gastrulation this overlap was just seen in 1 to 1 cells. So the number of cells that's showing overlap between OPA and OC drops down. Again, single cell RNA sequencing data for OPA and OC again confirm our previous data. And confirm that OPA and OC overlap is just before gastrulation and for a very short time. Here we investigated the dynamic protein binding to the regulatory elements of head-specific gene using ChIP-seq data at different time points. Here we compared OPA, OC, ChIP-seq data with and Bicoid and Zelda ChIP-seq data together, and we can see that for example for this enhancer G30 this enhancer driving giants head expression at earlier stages, and we can see OPA, OC as well as Bicoid and Zelda could bind to this region. But if we check this enhancer, which is driving later stages of hunchback gene expression, we can see that just OC is binding to this region. So early enhancers are regulated by both OPA and OC, but late head-specific enhancers are OC only regulated. We use OCRNAi to study one of the important genes, which is hunchback expression. So we can see that when at earlier stages, with OCRNAi background, the hunchback expression is like wild type, but at later stages, its expression is stuck in the earlier stages. So our studies show that early enhancers can be regulated by early transcription factors, as we can see here, but at later stages just OCs bind to enhancers that are regulating gene expression, that genes that are important for head development. So, and in future we will be focusing on tissue-specific ChIP-seq analysis as well as single cell attack sequencing to further study the dynamic mechanism of these transcription factors in embryo brain development.